Next up, we have Dr. Cantillon from Cerecin, and we're very excited to see what they've been up to.
So thank you for inviting us. I think we were here once before. My colleagues, the Chief Science Officer, Sam Anderson, and our CEO, Charles Stacey. I'm the Chief Medical Officer and delighted to give you an overview of where Cerecin Pharmaceuticals are. We fit into this space, I believe, mostly because of the metabolic interest we have, but also in the broader sense, as I'll try and explain in terms of neurodegeneration and maybe body generation. Our target is the metabolic causes of neurological diseases. So we are a private biotech company focused on those metabolic causes of neurological diseases. We're actually the leading company in that area right now, bioenergetics and metabolism, with 3 late-stage programs in migraine prevention, in Alzheimer's disease, ready for phase 3, and we also have a pilot program in infantile spasm that I will speak to. We have leading financial institutional investors, a large part from Korea, SK Securities and so forth, you can read there. We also have long-term Fortune 500 investors. Our major investor was the Nestle group, as well as Wilmar. We have global offices to optimize these clinical trial operations and also R&D labs in Singapore, USA, and Australia. And we have an experienced team of CNS drug developments from biotech and big pharma. So, as I said, our science and our pipeline are focused on the intersection of neurology and metabolism. So we call it neurometabolism, and try to just sketch it out here in people's minds. So CNS is a broad area, neuropsychiatry, of which neurology is at least a big part. And on the other side, we've been talking about various pathways, ketogenic and others in the metabolic sphere. And where those two come together, we believe is where there's a lot of opportunity right now, which is close to hand in the 3 areas that we're developing for our first compound, CER001 tricaprolin, in those 3 indications. And we also have a follow-up compound that I can't say too much about yet, but we have a pipeline of other drugs in the same area that we've been working with for about 15 years. So that lead compound, tricaprolin, is being developed in the 3 different disease indications. And you can see that the most advanced is Alzheimer's disease, where we're about to go into a phase 3 study. And the second is in migraine prevention, also going into a phase 3 a little after that. And we have an ongoing pilot study in infantile spasms. The first 2 should be ready for an NDA, a new drug, by near the end of this decade. And the infantile spasm, which is a shorter, smaller study, may be shorter. We'll see. The ketone marker that we're just starting to work with may have very broad indications, both within this neurometabolism, but perhaps also in the same areas of ketones interest, which would be the heart, the renal conditions, musculoskeletal, and so forth. And we also have some other precursors and other ketone drugs in development. So a little repetition, maybe if people weren't at the session yesterday afternoon. The whole idea of ketogenesis is highly conserved, meaning you see it in all species, basically, as a physiological process, because it's almost like a backup. It's produced under conditions of low glucose availability, such as fasting, or could be ketogenic diets. It's a metabolic state, ketosis, where glucose scarcity or diet and consequent reduction in insulin resistance results in hepatic production of ketone, as you see on the drawing here, coming out, the ketose, which is an alternative energy for the brain, the heart, the voluntary muscles, and other tissues in the body. I mentioned even kidney, we're discovering, and others. And as such, you can produce up to 60% of the brain's energy need. And the brain, of course, is a notoriously greedy organ with only essentially 2% of weight and still demanding 60% of brain energy. And so one can see that there's an evolutionary connection between nature producing this conservative process in order to be able to protect the CPU or the brain. The production of keto bodies, we know, is suppressed by a carbohydrate diet, which is often actually a lot of the general diet, especially in the West. And in terms of numbers, after an overnight fast, ketones generally would supply about 3 to 5% kind of in a general population before you would start to do any specific either diet or treatment work. And I think you can see what I've said there on the diagram on the right-hand side. So where do ketone bodies then as a treatment or giving ketones come in? Well, they act as an energy substrate, but more than that. So divided up these ketone bodies that we would be giving as a medicine into either the energy pathway or in cell signaling. So the energy we've just talked about, the way that energy is produced is when the BHB is converted to acetyl-CoA and into ATP in the mitochondrion, so-called battery of the body and especially the brain. The cell signaling is probably more interesting for a variety of reasons, but is much less right now with in terms of research, but it's coming on very fast. So you see FFR and HCAR, and also going through BHB through histone deacetylases to the nucleus, so that essentially you have gene expression changes. So there is a wide range of direct and indirect signaling that have numerous downstream effects, whether it's that nucleus effect that I just spoke about. There's also interesting changes in lipid metabolism, sirtuin activity in the NAD, and also sympathetic tone. So there's a whole body of work looking at inflammasome. So consequently, ketones as such, either externally or through this mechanism of diet, can have an effect on many neurological conditions, such as seizure disorders, and that's probably the oldest and well-known, but also migraine, ischemia, traumatic brain injury, Parkinson's, Alzheimer's, and glutamate toxicity. So our compound, tricapelin, leverages this pathway, providing an exogenous source. And one of the reasons for that is it's very hard to have a diet all the time. If you have a very severe epilepsy and need to do that, you have to be very careful of maintaining that balance and also having your nutrition. And by giving tricapelin with a lot of the same mechanisms of action, you're able to at least not have to go to such an extreme with your diet. So essentially it's given in orally instead of being released as it would be in a diet from the fat cells and through the liver very quickly. Within basically 30 to 60 minutes, the ketones enter the bloodstream and supply the brain as well as extrahepatic conditions. Now, as I said, this builds on decades of use of ketogenic diet in treating multiple drug-resistant neurological conditions. And even before 1923, when the Mayo Clinic published on the treatment of epilepsy, it was known right back to the Greeks and Romans that various forms of diet could help certain conditions, including seizures. But now it's beginning to be more scientifically understood why. That essentially ketone production comes through either this diet or starvation, or from the outside, as we're going to talk about. The classic keto diet is typically composed of a macronutrient ratio, 4:1, 4 grams of fat to every gram of protein, plus some carbohydrates combined. And studies show that while it's effective, it's extremely challenging. So I've just gathered here some reviews that you can also look up. One of them is to do with Alzheimer's disease, 11 animal studies, and I think it's 14 now, human studies concluding that ketogenic diet is a promising way to alter cognition, short-term memory, focus, and attention. Also, that there are a whole series of newer studies on various types of epilepsy, especially those where they've had drug resistance, and finally on headache and actually other kinds of pain, showing the efficacy there. So we're going to use that in our tricapelin pathways in terms of bringing these to being an actual drug treatment, allowing the plasma ketone levels to be raised to the level you see through these older methods of diet and so forth, and allowing patients to maintain a healthy, normal diet. So this is what, on the left, an ideal drug might look like. On the right is our tricapelin profile, and on the all the way right is what it looks like, an attractive liquid formulation of the drug. So you want to have, in an ideal drug, an oral administration. It should also taste nice for convenience. This has a vanilla taste, which is quite pleasant. It needs to be able to add it to standard of care, which we have tried with this drug. So in the Alzheimer's case, it would be with standard treatment like donepezil, Namenda. Good safety and toxicity, which we've had both with our formulation drug as well as in the background literature. It should have quick improvement. And I mentioned that you can actually see effects within the hour, but you also have sustained benefit in the standard FDA required outcomes for Alzheimer's disease, like the ADAS-Cog. It also has to have a reasonable price, and we think we can deliver that. And finally, it should not need any external monitoring. So unlike some of the other Alzheimer's treatments that are just being approved, there's no MRI needed, no blood measuring, no brain scans and PET scans and so forth, because this is very safe and can be used in that way. So I think we've talked about Alzheimer's disease the last couple of days, but just to remind us, is one of the major diseases of aging and particularly disabling. And it's not just the patient, but it's the whole society, caretakers and families. There's been estimates up to 300 billion in total costs with worldwide 50 million patients. Aging is the single largest risk factor. And this is going to increase because of the aging population to nearly double within the next 25 years. The current treatments do provide only symptomatic relief. The current ones give about six months benefit, and then they wear out. They just don't have any further. And the newer approved antibodies are showing benefit, although it's some controversial about actually how much they actually show on a scale after 2 years, and you do have to do brain imaging and some other mechanism. So tricaptan is a novel mechanism, and it is going into late stage phase 3 studies, as I said. Numerous amyloid and tau-based treatments have failed, and we believe that this, even if those are approved, will be a distinct and attractive drug class. So I'll go through a couple of the trials that have been done already in mild moderate disease, showing immediate and sustained effects. And this trial that I'm speaking about, which is going to begin next year, is across these countries. Essentially, we want to get a global representative population. So one of the interesting studies shown was correlating this glucose metabolism defect that occurs in Alzheimer's disease. And you can do this with a PET scan. It's actually one way to diagnose Alzheimer's disease. We used to do maybe 20 years ago, and it's still current today, that certain areas of the brain, as you see here, are very specific for Alzheimer's and distinguish it from some other conditions. And that low blood flow can be detected decades before the actual clinical signs of dementia. As you can see here, some studies showing 25 years beforehand. That decreased glucose metabolism is seen in people at risk for AD, and some studies even go back to age 30. So we used tricaptan in a first study, which was just a crossover single dose, and resulted in a rapid improvement in the cognition. And 20 patients, you can see the design. It essentially elevated both serum ketone bodies and this ADAS-Cog standard outcome. And you can see on the extreme right that correlation. Paragraph recall is one of the best short-term memory tests, and essentially as high as you can go is higher you're seeing in the paragraph recall change scores. We did then a second study looking at a change from baseline in a longer period of 3 months. And again, you can see that it's in a mild moderate stage of disease. What was interesting in this is that it was most prominent in ApoE4 negative, which is in different populations, a little bit less than half of the Alzheimer's patient population, but other studies have shown it was in the ApoE4 positive as well. So that's one area that's still under some review, but you do see that there was clear evidence, not just short term, but lasting up to these, at least in this study, 3 months versus placebo.
Mark, just about 1 minute, please.
I'm sorry?
Just 1 minute left. Yeah. Can you wrap it up, Mark?
Yeah. Yeah, so let me go to the study we're doing, which is this. We're going to do that over 6 months and hope to show a difference between the two drug treatments. We also have some evidence of efficacy in migraine and presenting that data in the coming months at different meetings. And we have data in infantile spasms, where you can see from the rat model as well as in humans that there is improvement. These are our partners, different pharmaceutical companies, and this is our board of directors, people who also have been in the biotech and pharma space, and these are our different offices around the world. I'll end with that on time.
Thank you so much, Mark. This is
Thank
pretty exciting stuff, and best of luck in that phase 3 trial. We're all watching that with great anticipation.
Thank you.